Good morning everyone, myself Amima Mallik, and today I will be explaining about the mechanism of nucleotide excision repair, which is a type of DNA repair. So this mechanism plays a crucial role in maintaining the stability of our genetic material by repairing the damaged DNA. So in this topic and in this PPT, we are mainly focusing on the repair mechanism in prokaryotes. So let's start. So let's understand the basic importance of DNA repair. Our DNA that can suffer from various accidental damages due to environmental factors like UV light or chemicals. These damages, if left unrepaired, can cause many serious mutations and can lead to many serious malfunctions. Thankfully, our cells have sophisticated repair mechanism to fix these issues. So one of this is nucleotide excision repair. But in this topic, we are mainly focusing on the prokaryotes. So what is nucleotide excision repair? It is a key pathway to fix large DNA damage like damage from UV light. It detects the distortion in the DNA double helix such as thymine-thymine dimers or bulky additions as DNA base. These damaged part is removed, leaving a gap which is further filled by DNA polymerase. So to begin with the mechanism of nucleotide excision repair, we need to keep in mind some certain proteins. So the proteins that are involved in this mechanism are UVRA, UVRB, UVRC and UVRD. UVRA and UVRB forms a complex to find the damage, whereas UVRB and UVRC makes cut on the both sides of the damaged area. The UVRD unbinds and removes the damaged segment from the DNA. So first step is the recognition step, where the UVRA recognize the damaged site in the DNA such as the thymine-thymine dimers, which is caused by the UV light. UVRA binds to the site of the damage, which means UVRA recognize the damaged site and binds to the thymine-thymine dimers. Once the UVRA binds to the thymine-thymine dimers, simultaneously the UVRB creates a bubble which binds to the DNA because of the affinity of dimers, which leads to the conformational change in the DNA. So the conformational change is caused by the UVRB leads to the release of the UVRA from this DNA. So the next step can be called as the recruitment of the UVRC. So the UVRC is recruited to the site. UVRC is responsible for making the incisions on both sides of the damaged DNA. So UVRC makes a cut, whereas in the 4th, 5th nucleotide downstream, which is a 3' and 8th nucleotide upstream, which is a 5' of the lesion. So the next step is excision. After the incisions are made, the damaged DNA segment is removed from the site, and then the new enzyme is introduced in the site. So the new enzyme, which is a UVRD helicase, which helps in unwinding and removing the damaged DNA segment by breaking the hydrogen bonds between them. So the next step is DNA repair synthesis. A new patch of DNA is synthesized to fill the gap between the removed DNA. DNA polymerase 2 fills the gap by synthesizing the new DNA, and finally the DNA ligase seals the new strand into existing strand, completing this process. So this was the overall mechanism of the nucleotide excision repair. Let's understand the advantages and disadvantages of this process. So the advantages includes of the broad specificity. It has a genome stability, which means it helps in maintain the genome integrity by removing lesions that can cause mutations if left unrepaired. For the broad specificity, NER can recognize a large pair of lesions, including the bulky adducts, which is formed by the pyrimidine, that is the thymine dimers. The disadvantages of this nucleotide excision repair, that this particular process requires a significant amount of energy, enzymes and proteins to identify and excise the damaged DNA segment and synthesize new DNA. It is also time consuming, as it is a multi-step process. It is little slower than the rest of the repair mechanism. Yeah. Thank you so much.